Hello, everybody. My name is Boris Heifets. I'm an anesthesiologist and neuroscientist at Stanford University. And I'd like to thank you for inviting me to the FAIR Medical Student Anesthesia Research Fellowship Virtual Summer Program. I'm going to tell you in the next 10 to 15 minutes a little bit about what I'm doing in the lab, and I look forward to meeting you online and answering some questions. So for the past several years, I've been collaborating pretty closely with psychiatry. And if you're like most of my colleagues, you may be wondering, what on earth does anesthesia have to do with psychiatry? And so I'll just start by saying this, that the most powerful psychotropic drugs in the hospital are actually in our drug carts. And the more we find out about how these drugs work and what they're capable of, the more obvious it is that we may be able to influence outcomes, patient outcomes that extend far beyond their brief stay in the operating room. One of the best examples of this phenomenon is probably ketamine, which some of you may be familiar with. And in addition to being a sedative and an analgesic, it's also a pretty powerful antidepressant drug, which has received quite a bit of press in the last couple of years and has recently gotten FDA approval as S-ketamine, which is one of the enantiomers. Anyway, that's really the starting point, is that these collaborations with psychiatry really have grown out of this idea that we can really start moving the needle on patient outcomes and just understanding basic mechanisms of the brain by upping the ante of the kind of interventions we're willing to do and capable of doing with appropriate monitoring, et cetera. That is more typical of the anesthetic specialty. So the broad picture here, if I can advance my slide, as I just mentioned, is that there is a host of drugs actually beyond just ketamine that are really racing through the FDA approval pipeline in the last several years. And what's really remarkable about these compounds, and I like to think of them as a class, but really they're quite distinct, both in terms of their pharmacological mechanism and the disorders they're meant to treat. But they are they share a unique property in that they act almost immediately. They have a powerful immediate psychotropic effect, and their effects last far longer than their levels in the blood. And there are really not that many examples of medications like this. So in the case of MDMA, which I've done a lot of mouse work on, MDMA given once or twice in the context of psychotherapy can trigger years-long remission from severe post-traumatic stress disorder. That's now in phase 3 trials. Psilocybin, the main ingredient in magic mushrooms, is now being investigated for major depressive disorder. And again, this is just a single dose. And as I mentioned, S-ketamine has been approved for depression. So this sounds great. Maybe we should just stop there. The premise of my work really is that these drugs are a great starting point and provide a strong signal, but they're not perfect by any means. And each one of them comes with a whole host of unintended side effects. And this is true if you look at any other field of medicine, whether it's cardiovascular medicine, chemotherapy, or certainly anesthesia, that anesthetics have just gotten better and better because we've honed in on the therapeutic factors that make them work. So, MDMA is a drug of abuse. Psilocybin is clearly not appropriate for every patient. And S-ketamine and ketamine are actually also, probably not the kind of thing you want to send home a patient with. So how do we address this in the lab? And it relies on a, pretty vertically integrated pipeline that's really taken years to develop, and at least in my hands, and we're still, we've only scratched the surface. So the first thing, if we want to go bottom up, is we model drug effects with mouse behavior. Now, mice aren't people, but we can learn a lot because, again, they're mammals, and a lot of this neural circuitry is conserved. Now, using some of the most advanced tools in neuroscience, we can perform very precise manipulations of activity in specific brain circuits. We can then move forward from there and identify synaptic and circuit mechanisms, potentially identifying receptors or brain stimulation targets that we wouldn't have thought of just based on the identity of the lead drug. And then finally, we can take those ideas and test them in humans in mechanistic clinical trials. And again, hopefully feed that back into a virtuous cycle of developing better medicine with fewer side effects. So I'll share briefly with you just an overview of what we did with MDMA. So MDMA is also known as ecstasy, has been actually demonized drug on FDA Schedule 1 since 1985. And for good reason. It's a party drug. One of the first uses of it before it was put on Schedule 1 is actually as an adjunct to psychotherapy. And that is really due to a very unique effect it has on what we term prosocial behavior, or this feeling of empathy and social connectedness that people feel while they're on drug. And therapists have used that to tremendous therapeutic advantage. So, as I mentioned, there are these twin sides, this prosocial and this drug reward effect. And my project basically was to see if we could split those apart. That's the first step in drug development. And the first thing you have to do is you have to model these complex human behaviors in a mouse. Now, this is really not the forum for me to get into those details, although I'm happy to elaborate. But basically, we were able to model something that really has all of the same hallmarks of human prosocial behavior in a mouse. We're also able to model drug reward and addictive-like phenotypes in mice. And we're able to do a lot of other instrumental learning, Pavlovian learning, and various different behavioral tasks that have been developed over many years to really suss out the various effects of MDMA. So once we have this behavioral model set up, as I showed you a slide ago, the next step is these higher end kind of precise manipulations. So again, now that we've been able to model it in mice, and again, our goal is to try and separate out what is it that makes MDMA prosocial and what is it that makes it addictive, and can we separate them in such a way that we can trigger one of those effects, the therapeutic one, without the other. So what do? Well, we have a combination of viral mediated gene transfer, optogenetic techniques, chemogenetic techniques. These are things that basically allow us to have millisecond control of genetically defined populations, meaning if I wanted to just stimulate the terminals of every serotonin neuron that projects from the dorsal raphe to the hub of reward in the brain, the nucleus accumbens, I can do that with millisecond precision. These are tools that have been developed in the last 10 years, many of them in Stanford. So again, we combine these kinds of manipulations with behavioral models that I just described. We do imaging, we manipulate circuits, and basically we're able to get a clean mechanistic understanding of how this drug is able to produce a fairly complex behavioral effect. And again, that's beyond the scope of this discussion, but we were able to just trigger that prosocial effect without the addictive effect. And this has now led to a number of clinical trials that are in very early stages of planning, and potentially some companies that are being spun off. So that's it's kind of an overview of how we went bottom up. And I want to just point out, that this is a process that can go backwards. And this is where having a fluency with basic science and clinical research is so important. And it's not necessarily that you need to be, leading a lab of both types. I mean, I'm not sure this is really going to work out the way what I'm trying to do, but it is important to have that kind of understanding of the language of basic science and also clinical medicine. And from the clinical side, we've tried to run this process backwards, basically, starting with an effect, a mechanistic finding in humans, and seeing if we can go back and model that in mice. So this really involves ketamine. And again, ketamine is, as I mentioned in the beginning of my talk, it's a increasingly widely used antidepressant. It's thought to act primarily via the NMDA receptor. It turns out that is not really the entire story. We published a paper a couple of years ago where we showed that naltrexone, which is an opioid antagonist, could completely block ketamine's antidepressant effect, which is a little bit surprising. It doesn't mean NMDA receptors aren't involved, but it points to a potentially new therapeutic mechanism, or basically a mechanism that people hadn't really thought about for antidepressants. So that's cool. Now let's see if we can model the same kind of thing in a mouse and really get some traction on how do you then redesign ketamine as a drug that doesn't have its kind of abuse potential and some of the dissociative effects, but still maintains its antidepressant effect. So this is just clearly too much for me to cover, but the bottom line is we're modeling ketamine and opioid receptor interactions in mice, and we're using some rather complicated genetic tools to basically label every cell in the brain that's activated when mice are on ketamine versus ketamine plus naltrexone. And this is really one of my favorite, this is just came out from my lab a couple months ago, but this is the first map that we've made using a combination of imaging techniques and this genetic, this genetic mouse model to illustrate, three-dimensional maps of what is the ketamine naltrexone interaction. So again, hopefully this will lead to circuit manipulations and perhaps drug discovery efforts or brain stimulation strategies that could treat depression without triggering some of those untoward effects of ketamine. So how does this relate to clinical practice? So what I'm describing for you here really is rapid interventions for psychiatric disease. Now, if you've spent any time in a pre-op clinic in anesthesia, you know that we spend a lot of effort trying to optimize patients from a cardiopulmonary status. So we have all these diagnostic tests, and we have all these therapeutic interventions to really tune a patient up prior to surgery. When it comes to psychiatric risk factors, which I should mention are really just as, carry just as much risk as some cardiopulmonary risk factors, we have patient history and really no intervention. So we haven't really been able to intervene on the time course that you need to prepare for surgery, which is maybe a week. These approaches that I've described to you with these rapid acting interventions like ketamine, like MDMA. MDMA is not ready for perioperative use by any means, but ketamine already is used. Can we target vulnerable patient populations and improve outcomes related to psychiatric comorbidities? So this is a clinical trial that we've started to address that very question. And what we want to know is ketamine has been used for years now as in outpatients and awake patients, but we have no idea if we can give it in the same way, the same dose to patients under general anesthesia and get an antidepressant effect. So we're looking at trying to enroll patients who have moderate to severe depression who are presenting for non-cardiac surgery. That's surprisingly, there are a number of patients who are essentially debilitated by depression that are still having surgery. And we're taking those folks and we're allocating them to either ketamine or saline. And then we're looking at this is a composite mood rating at 1 day after surgery. And this is a common endpoint for psychiatry trials. And we're also looking at opioids. So I hadn't done a clinical trial before, so we wanted a little sneak peek, make sure we weren't doing this completely wrong. So we took the first 5 patients and we did an open label study. These aren't randomized, and this is really just to optimize procedures. And what we see is just in these 5 patients, this is their depression score, and this is before and after ketamine. There's a pretty robust response on average, and 3 out of 5 patients would meet clinical response criteria, which is about what you see in outpatient populations. So that's promising, and it suggests that we may be able to see a signal here. We also looked at some EEG, which again, going back to how can you spin this into a better drug? So we're collecting information on mechanism, and we see some relationship between the extent of mood change and the shift of one of the primary frequencies in EEG. Again, for another time. Before I wrap up, I just want to give a really brief overview of basically my career trajectory, just so you can get a sense of how did I get to this point. So I started residency in 2010 after my intern year. 2013, I started 3 years of fellowship, which I really don't recommend, but that's what I did. A year of it was just research fellowship, and then 2 years was neuro fellowship spread out, basically 20% time over 2 years. And right around that time, I got a FAIR grant, and then in 2016, I became an attending. Then I got a KO8, which was wonderful. And then in 2018, I was appointed to a faculty position. And last year, we started opening the lab, a new lab. Of course, COVID hit, and then, you know, all hell broke loose. But anyway, so again, some grant support was key in that, you know, right early, right often. And I just want to point out, it took a really long time in order to show productivity for me to really get these projects off the ground. And it's really important, if you want to do this kind of work, to pick your departments wisely. And I have a couple words on that at the end. So that's really, you know, there's about 2013 to 2017 was a kind of a big dry spell for me. But again, it paid off, and without continued support from my department, I don't think I would have been able to do any of this stuff. I should also point out that basic science is hard. This is how long this mouse project took with MDMA, 5 years. The clinical project was done within that time. It started and finished while I was still doing a mouse project. And these are all the things that I didn't get. This is just a sampling of all the grants I applied for, travel awards, etc. You just got to get used to this kind of stuff. It's a numbers game, unfortunately. So what I want to leave you with, this is a blog entry I wrote for ESAS, which is the early stage anesthesia scholars. This is a community I helped found a few years ago, and there's a website. I wrote this piece basically just as some words of advice for what to look for if you think you really want to pursue research, any significant amount of research in your anesthesia career. So there's obviously a lot of people that made this happen, and this is really kind of the big message is this kind of work requires an enormous amount of support and a lot of failure before there's success. So with that, I want to thank you for your time and attention, and I look forward to meeting you all.